All right, so get ready because today we are taking a deep dive into a research paper with the potential to completely upend how we think about memory. We're talking about maybe not just forming memories, but actually targeting and disrupting them way after they've kind of settled in.
And the really fascinating part is this research isn't just theoretical. Scientists at the Scripps Research Institute have made some pretty incredible breakthroughs.
Yeah, this is straight out of Eternal Sunshine of the Spotless Mind territory, right? We're going deep into their work on something called, get ready for it, selective retrieval independent disruption of methamphetamine-associated memory by actin depolymerization.
Catchy title. Right.
Super catchy. But seriously, trust me, the findings are even wilder than that title.
Absolutely. I mean, for the longest time, we thought of memories as being, relatively fixed. Imagine a file saved to a hard drive. Once it's there, it's pretty much set. But this research suggests that some memories, especially those tied to experiences with drugs like methamphetamine, might be way more dynamic than we ever realized.
Okay, so less like a file on a hard drive then, and more like, a constantly running app on our phone, something that needs upkeep and energy to keep going. That's already blowing my mind. But where does the whole disrupting memories thing come in?
Well, that's where things get really interesting. This research is all about a protein called actin. You might remember it from biology class. It's famous for how it helps our muscles contract.
Oh, right, like how our biceps get all pumped when we lift weights. What does muscle building have to do with memory?
So it turns out actin is a major player in the brain too. Inside our brain cells, actin forms these dynamic, always changing structures. Think of them like teeny tiny scaffolding systems that help memories take shape and hold on over time.
Hold on. Our brain cells are literally building tiny structures to hold our memories. That's wild.
It is pretty wild. And get this research discovered that memories connected to methamphetamine seem to rely on a specific type of actin that's constantly cycling. It never really settles down, always on the move.
So most memories are okay, we're good here, let's just chill. But these meth-linked memories are nope, gotta keep moving, gotta keep changing.
Exactly. They just constantly restructure themselves. And the researchers found that this nonstop cycling makes those memories surprisingly vulnerable. It's like they have a built-in weak spot.
So while my memory of what I had for breakfast this morning might be pretty stable maybe, depends on the day. The memory of a meth experience is more fragile because it's built on this constantly shifting foundation.
Exactly. And that's where the possibility of disruption comes into play. By zeroing in on this specific type of cycling actin, the researchers found they could disrupt, even basically erase, those meth-related memories.
Wait, hold on. Erase them. Completely gone. How did they even test that? We're talking about memories here, not deleting files.
It's pretty amazing, right? They used this clever experiment called conditioned place preference, or CPP for short. They basically trained rats to associate a specific location with receiving methamphetamine.
So the rats learned to connect place A with that meth feeling.
Right. Now here's the really interesting part. Days later, after that memory was solid, the researchers injected the rats with something that disrupts actin, but only that specific type of constantly cycling actin we've been talking about. And get this, after they messed with the actin, the rats totally forgot they even liked that spot anymore. It's like the memory of that meth experience just vanished.
Wow, that's kind of mind-blowing. But how can they be sure it was messing with the actin that made the memory disappear? What if something else was going on?
You're right to be skeptical, and the researchers were one step ahead. To really lock down the link between actin and these meth-related memories, they ran another experiment. This time, they focused on this molecular motor called myosin II.
Myosin II. Okay, quick refresher. What exactly do molecular motors do again? It's been a while since bio class.
Think of myosin II like, a cellular mechanic maybe? It works in the background, tinkering away, interacting with the actin to control its movement and how it behaves.
So myosin II is like the stage manager, making sure the actin scaffolding is set up right for those meth memories to take hold.
I like that. And what they found was when they messed with the myosin II, it had the same effect as going after the actin directly. The rats' meth-related memories were disrupted even though they weren't actively trying to remember them at all.
Okay, so two different ways of messing with this actin scaffolding, and both have the same outcome, bye-bye meth memories. That's pretty strong evidence that they're onto something huge here, right? But let's zoom in even closer. How do we get from these tiny little proteins and cellular mechanics to an actual memory in our brains? It all seems kind of abstract.
That's the million-dollar question, isn't it? How do these microscopic things turn into something as complex as a memory? To get that, we gotta visualize how memories actually get physically encoded in the brain.
Hit me with the neuroscience.
Okay, so imagine your brain is like this massive network of roads and highways all connected. Each brain cell or neuron is like a city, and those connections between them, those are the synapses, the spots where information is exchanged.
Okay, I'm with you so far. So instead of memories being like files stored in a specific place, they're more like the patterns of traffic flowing through all these roads.
You got it. And just like adding extra lanes to a highway can make the traffic flow better, changes to the connections between neurons making existing ones stronger or building new ones, that's how memories are physically, like, written down.
So the more we use a memory, the busier that highway becomes.
Exactly. And that's where those dendritic spines we talked about earlier come into play. Remember those tiny little off-ramps on the neuron highway?
Yeah, the ones that actin helps to build.
Right. Those dendritic spines are like the prime real estate where synapses form. And they're super dynamic, always changing shape and size in response to what's going on.
So our brains are like constantly building new off-ramps and expanding the highway system to fit new memories and make old ones stronger.
You've got it. Now, what these researchers saw was that in the rats who formed those strong meth-related memories, the areas of their brains connected to those memories actually had way more dendritic spines.
More off-ramps, more connections, basically making those meth-related pathways super strong and easy to access. But here's the big question. When they messed with the actin, did those extra dendritic spines just, like, disappear?
You're catching on fast. That's exactly what they found. Disrupting the actin was like they went in and bulldozed those extra off-ramps, making it way harder, maybe even impossible, to access those meth-related memories. Like the memory was still out there somewhere, but the path to get to it was totally overgrown.
That is wild. It's like we're seeing memories physically happening in the brain, and even crazier, we're starting to figure out how to tweak it. This is huge, right?
It really is. But we got to remember, this is still early days for this research. This study looks specifically at meth-related memories. In rats, we need more research to see if these findings hold true for other types of memories. And of course, in people.
Right, gotta be careful about jumping from rats right to humans. Everything might not be a perfect match. But still, the potential here is massive. Like, imagine being able to help people struggling with addiction by going after those deep-seated cravings and triggers.
It's a really exciting idea. But just like any powerful tool, we have to think about the what-ifs and the ethical side of things, too. The ability to change memories, even for good reasons, that's a big deal.
It really makes you think. Like, where do we draw the line? If we can erase the memory of something traumatic, does that mean the experience itself didn't matter as much? And what about just erasing memories because we don't like them, even if they're not really hurting us?
Those are the tough questions scientists, ethicists, everyone really needs to think hard about. Having these conversations at the same time as these scientific discoveries is crucial. It's the only way to make sure these findings are used responsibly. This isn't some sci-fi thought experiment anymore. It's a real conversation we got to have right now.
I hear that. We've gone from memories being, like, set in stone to maybe being able to reshape them. That's a lot to take in. One thing's for sure, this research has opened up a whole new world of possibilities and some big, big questions about what memory even is.
Definitely. This research gives us a glimpse into how incredibly complex and dynamic the brain is, and it highlights how much we still don't know about memory, addiction, all the things that make us who we are.
I like that. Well, deep divers, we've covered a ton of ground today. We explored some seriously groundbreaking research that turns our understanding of memory on its head, dug into how the brain actually works, and even waded into the ethics of messing with memory. What a ride.
It's been quite a journey, and honestly, we've barely scratched the surface. There's so much more to find out about this stuff.
That's what makes these deep dives so awesome, right? If you're as mind-blown by this research as we are, definitely check out the show notes for a link to the original paper. It's well worth a read. And hey, if this got you thinking about memory, neuroscience, all that good stuff, hit us up. We love hearing what you think. Maybe we'll even pick apart some of your questions on a future episode. Until next time, keep those brains firing, and remember, the journey is just as important as the destination.